I am a product manager at MilliporeSigma, product manager specifically for custom immunoassays, and I wanted to share with you our custom immunoassay services and a brief case study just to give you an idea of what we can do at MilliporeSigma and some of the types of projects that we take on. So our tagline is basically, we hope if you dream it, we can multiplex it. And to do that, we realize that everybody's research is different. Everybody is looking at biomarkers for different applications in different sample types. And we can't possibly offer an off-the-shelf assay that meets everybody's needs. I mean, we try as best as we can. We try to do research with our customers. We try to group things into panels that we think will be useful. But in the end, everybody has unique needs, and everybody has biomarkers of interest that they want to measure in their sample. So at MilliporeSigma, we've come up with a custom assay services program where we hope to address some of your needs in terms of measuring things that are exactly for your sample types. What this looks like is, so it kind of comes in two flavors. On the left, we have our very basic custom assay service program where we can combine things from different panels to meet your needs. So, perhaps that human cytokine that we group in our immunology panel is necessary for cardiovascular research, or in this case, sepsis, metabolic, and cytokines in one panel. So we offer a service where we can combine analytes from 3 or less panels with fairly quick turnaround and our standard validation services. And we call that our special product release program. I think what's more exciting though lately is we've been, as the fields of immuno-oncology, cancer research are evolving so quickly, there's markers being published almost every day that people are interested in measuring. And when it takes a year to launch a panel to the validation and specifications that are required for a multiplex assay, we thought we would offer a service where we could develop completely new assays for you from scratch. This would include developing new analytes, multiplexing and verifying those new analytes, perhaps with existing analytes, or testing them on your samples. In addition, we realized that when working with CROs and pharmaceutical companies, the level of validation that's required for your assay is different from customer to customer. So we offer a menu of validation services. And I kind of broke it down in this graphic for you just to kind of summarize our process. So we like this to be very iterative, and it involves consultation with scientists. So we would initially sit down with you and do client feasibility. You would give us your list of desired markers that you would like to measure, your sample type and your species, and we would run through that for you and give you a feasibility analysis. Second, you would then work with our team to kind of formulate a work plan that's right for your project. And if you decided to proceed with the order, we would go in a milestone-based approach, where we would first develop any new analytes, then multiplex and verify them, and then produce an assay that's controlled for lot-to-lot consistency. So we operate in the kind of this milestone-based approach that you may be used to utilizing if you're in a CRO or a pharmaceutical company and outsourcing assay development. I'll share with you some key program statistics because we get asked this question quite a bit. This is our custom assay program, as you see it here, has been in existence for about 2 years. We've done everything from a single-plex assay to a 48-plex that we've developed from scratch for a customer. And this is just including new analyte development projects or new assay development projects exclusively. The average plex size seems to be about a 12-plex. That's where we see most customers who are looking to multiplex on the Luminex platforms. That's what we see as most common. We've done over 50 new analyte projects, and we have a success rate of new analyte development of greater than 98% right now. You may say, well, why not 100%? We do our best to give feasibility up front, and there are some projects which we deem technically challenging, but a lot of clients will want to proceed anyway. And so sometimes, unfortunately, science is science. We can't always come up with a solution, but we try our best, and we give an upfront feasibility, and sometimes clients want to proceed and try it as well. So that's why we probably have a success rate of below 100%, but greater than 98%. I'll give you one example of a customer we recently worked with. This has been published in an online article If you stop by our table later, we can share it with you and email you a PDF copy. This customer is actually at Bayer, and they came to us with two very unique projects. The first was where they were running an assay that I believe had 6 individual ELISAs. And Bayer, this site for Bayer is interested in looking at endocrine disruption. So this is important for whether you're developing a therapy or you're doing crop science research. This centralized facility had an assay that they had run and had developed in an ELISA format to basically measure endocrine disruptors. It wasn't practical to use 6 individual ELISAs, and they came to us and said, you have most of the analytes we'd like to measure, but can you develop a new one, make it a 6-plex, validate it on our samples, and sell us the assay? Of course, we said yes, and we proceeded with the development of the project. As we were working on that project, a second project came up where Bayer asked us to re-optimize a standard curve for measuring hormones. So, not all customer samples are the same, and when we develop assays, we always like to have the customer sample in-house so we can test it. But sometimes, customer samples, whether the patient is diseased or, undergoing some sort of treatment, levels of proteins can be much higher than we're used to dealing with, and that may require re-optimizing the standard curve or the range of the assay. So we took that on as a second project specifically for Bayer. Just a very high-level overview of the project since I only have a couple of minutes left. How this project looked, in step 1, we developed new analytes. In this case, it was testosterone. This was actually a little bit challenging because there's some regulatory requirements involved with storing this protein. So, I guess in theory, you have to worry about people injecting this into themselves, maybe if you're a bodybuilder or something. So we had to kind of get around that, but we worked very closely with Bayer on that. And then we multiplexed these analytes together and verified the assay. I mentioned we like to use customer samples. That's always ideal. In some cases, our customers are located in countries where they cannot send us samples, so we work up a strategy whereby we will send them iterative alpha and beta kits during the development process for them to test on their samples in-house. It's always easier if they can send them to us. This customer was in France. That was not a possibility, so we sent them beta test kits during the process, and they made sure that the assay worked for them. We went a step further and then compared this to ELISA. So Bayer gave us their gold standard ELISAs. As you know, if you've been running an assay for 5 years and that's what you're getting results with, and you change platforms, you have to make sure your results are consistent. Whether or not the multiplex assay works better or not, or is maybe giving you a truer measurement of the protein, you need consistency. So you can't run one patient sample and then use a new platform and have different results. So we compared this assay to the individual ELISAs that Bayer was using, and they were very satisfied with those results and decided to proceed. And we operate on a milestone basis, so all of these data were reviewed throughout the process. And then finally, we like to see the assay working in final format in the customer's hands before we send it to them. Custom assays are made individually for each customer, and there can be things that maybe we don't account for when we're developing the assay in-house, like what kind of software are you using, what kind of washer do you have, even what kind of magnet do you have. So we like to send them an assay in advance just to run before we do the final kit shipment. And that passed, and what we generated was this. This is the standard curve, just to give you an idea of what the assay looks like. It's now being sold commercially. Bayer was very happy with the results, and they released this to us to sell to other customers. So that's all I have. I just wanted to mention, if you're interested in any of this information, we have some literature at our table. We have a custom menu of services that describes everything we offer. For custom assay development, we can do anything from bead conjugation to full assay development, and we have quite a few of those projects going on at any one time. And also our analyte brochure, so we maintain a comprehensive list of every analyte we sell, not only on our online finder, but also a printed copy as well. Some people prefer print, so we always have that. And you can email us anytime. This email actually goes directly to me, and I work with a scientist to address feasibility on any custom project. You're also free to visit our website and fill out a form, and we'll get in touch with you pretty quickly. So I'll take any questions, and thank you for your attention.